All right. So up to this point, we've been discussing, we first started discussing what proteins are made out of. We've talked about different classes of proteins, enzymes and G proteins, and where they're located with respect to within the cell. But now for today's lecture, we're going to be talking about, we're not going to be focusing on a specific disease, but rather focusing on how we measure and analyze these different proteins. And we'll be talking about this within the context of, it's not kind of in the context within COVID-19, pregnancy, and anthrax. Three seemingly unrelated things, but all are related based on how we try and detect them. Because they all kind of share a similar detection method, and as I've mentioned in the past, there's like some aspects of this class that have kind of like stayed the same over time. There's an old cartoon developed by a pretty talented student that was in this class even before my time, where they're asking the student about studying for this class and learning about how the pregnancy test and the anthrax test use the same mechanism, and having a non-specific version of that test making you think whether either you're pregnant or you have anthrax. But yeah, since no one really found that funny, I might have to find a different cartoon. Even though that was forced, I actually appreciate that. Thank you. All right. So all three of those tests use the same mechanism. It's called lateral flow immunochromatography. So in the pre-lecture, you saw what chromatography is. Lateral flow meaning that it has a sample port that is then kind of elutes across laterally, and the immuno portion means that it's an antibody-mediated detection kit. And so whether it's detecting of anthrax in a sample like this, whether it's a urine sample for a pregnancy test, or as all of us, as I'm sure, are very experienced in now, a deep nasal swab for a COVID test, for an antigen COVID test. All three of them have the same mechanism. So one of the key methods that we use to detect and analyze proteins is called electrophoresis. Does anyone, whether you have a background in Latin or not, does anyone want to define for us what electrophoresis is? Why don't you take a stab at it? Yeah, that's pretty much right on. So the electricity, it's using electricity to promote the instead of draw it, I'll say movement. Phoresis, I don't have a background in Latin, so I'm sure you all might be able to have the exact definition of what phoresis is, but we usually think of it as the movement of proteins across an electric field. And so we use this technique of electrophoresis to try and typically separate proteins based on their charge and based on their charge and by their size. The way in which this works is we typically try and apply a positive charge to the bottom of the gel. And the reason why we do that is because you want the flow of the current flow is, it's typically the flow of electrons, and electrons are negatively charged, and they'll move towards the positive end. And so we create a positive charge here at the bottom to promote the movement of our protein samples down this gel. And so gel, even though seems like a fairly clear and a term that does not seem like it would be worth highlighting in blue, but what I want to try and identify and narrow in on here is that when we talk about a gel, we're talking about specifically a polymer network that's in some aqueous solution. That's what defines a gel here. And so what that means is there's this polymer network where there's a porosity that enables our proteins to move through. And we change that, altering that porosity is pretty key for enabling and how proteins can move through the gel, move through that polymer network. And it's in an aqueous environment. So I'm sure a lot of you, whether it's from the labs that work in our department or through all of the different readings you all have done in your spare time, you may have come across hydrogels, or if you've changed diapers, which has hydrogels in them. The aqueous environment that a hydrogel is in is water. And so it's a water swelling polymer network. And so in this case, it's not quite water. We use a different aqueous environment here. But the key thing to note is that the porosity of these gels dictate the ability of these proteins to move through them. And what will end up happening is, as we put them on top and as they run through the gel, they'll end up separating into bands. And you can ignore the color difference here. That color is just to indicate different proteins, but the rate at which they move is typically determined by their size and their total charge. And so you can imagine that the smaller proteins are able to travel more further deeply through the polymer network, and typically the larger proteins are at the top. And we typically are able to stain these proteins in order to try and look at their separation, because usually what we have is when we run a protein sample across a gel, what ends up happening is, if we don't do any staining or do anything to it, we call that a native gel. So when you take your native gel from there, you can either stain it with something, and we'll talk about that later, or excuse me, or you can do other methods of analysis that we'll talk about in next lecture. One of the most common forms of gel electrophoresis is called SDSPAGE. The SDS stands for sodium dodecyl sulfate. That's an anionic detergent. So what's really important here is a lot of our proteins, based on the amino acid sequence of the hydrophobic regions and hydrophilic regions, our proteins are folded in a certain conformation. We do a couple things here. We'll actually first try and denature the protein. Denaturing means basically trying to unfold it, unravel the protein, so that we can make it as linear as possible, so we can have every part of the protein available to bind to or to try and detect. So we'll first denature or unfold the protein. Actually, it's not on the slide, but what you'll actually do, if any of you have the pleasure of doing a gel electrophoresis, is you'll actually heat denature your sample. And so that's one step of unraveling the proteins to open them up. Another step is putting your sample in this sodium dodecyl sulfate, or SDS buffer. What's key here is that this anionic detergent will bombard every protein with a negative charge. Typically what we'll have is you'll have one SDS molecule for every 2 amino acids. And what ends up happening is the benefit of bombarding it with a negative charge is that now it can flow with the current, the electric current, towards the positive end. So that helps draw it down. But what we're doing here now is we're not separating it based on charge, because everything is going to have a negative charge now. So with SDSPAGE, the key thing to note is we're going to be separating it strictly by its size or strictly by its mass here. And so the next piece of the acronym SDSPAGE is PAGE, the polyacrylamide gel. Yes?
So that was all SDS, and the benefit of unfolding allows it to be bombarded with a negative charge. Is that what you're saying?
Yeah, I'm glad you asked that. So bombarding with a negative charge. So everything is going to have a negative charge, and negative by negative is going to repel one another to further open it up.
Okay.
So like the SDS accomplishes 2 key things here. It accomplishes providing a negative charge to the protein so that it can flow with the electric current across the gel. And it also further helps to unfold and denature the protein, because if there's a negative charge next to a negative charge, they're going to be repelling each other and helping to spread it out. One last thing I forgot to mention is we talked about in the very first lecture that some proteins have these disulfide bridges that cross-link a protein. And unfortunately, SDS isn't able to break those disulfide bonds. And so we have these reducing agents that'll break these disulfide bonds. One is called beta-mercaptoethanol. Typically, a rule of thumb is you can tell if someone's working with beta-mercaptoethanol because it smells like rotten fish. It smells terrible. But it's a really potent reducing agent that's able to break the disulfide bonds and further denature the protein. And so 3 key denaturing steps that we typically use for SDSPAGE is heating, the presence of SDS, and reducing agents to break disulfide bonds. The PAGE portion of the name of this method stands for Polyacrylamide Gel Electrophoresis. So we talked about what a gel is. We talked about in the previous slide what electrophoresis is. It's moving these proteins across an electric current down this gel. Polyacrylamide is the polymer network for this technique. It's really cheap and really easy to make, and 15-ish years ago, everyone had to like make their own gel and pour it. Now it's super cheap just to buy it and have someone else make it for you. But the key thing to note here is that polyacrylamide is the polymer network that makes up the gel in SDSPAGE. The buffers are typically have a high pH around 9, so trying to further create this environment that has a negative charge. And then what the key thing to note here is in electrophoresis, we typically can separate proteins based on either their charge or their size. With SDSPAGE, remember, the anionic detergent of SDS bombards every protein with a negative charge. So we're not separating by charge here. We're only separating by mass or size. And we typically, the units for that are kilodaltons. So daltons are typically grams per mole. And so whenever you see it, you'll have like a standard ladder that shows, typically up here, you'll say like, this is 200 kilodaltons, 100 kilodaltons, 70, 60, 50, 30. And so we separate by size or by molecular weight, and the units for that are kilodaltons. So here we have both structures for SDS and polyacrylamide. There is an aliphatic chain here with the highly charged sulfate group at the bottom here. And so what that enables us is that within hydrophobic regions, this fatty acid chain will enable it to embed itself and provide a negative charge even within the hydrophobic regions of a protein. And the polyacrylamide network is actually a combination of both acrylamide and bisacrylamide. What's key to note here is that typically bisacrylamide creates the cross links between different segments of the chain between acrylamide networks here. And just last thing I want to say, ammonium persulfate and TMED are important for the radical polymerization that promotes this network to occur. One last key thing is, or 2 more key things. So typically, if you increase the acrylamide concentration, you're making it less porous. So if you think about this, if you have, this marker doesn't work great. If you have more acrylamide available, you're creating a denser polymer network that's less porous. However, if you decrease acrylamide concentration, bless you, if you decrease acrylamide concentration, you have a more porous structure here, enabling, making it more easy for large proteins to migrate through. And so you typically have to try and, the key thing to note here is that it's important to be mindful of what your acrylamide concentration is when you're making a PAGE gel. The last interesting thing is that acrylamide is also a neurotoxin and it's also carcinogenic. So when you're working with the acrylamide monomer, it's important to wear gloves and to be very careful. Polyacrylamide in its polymer form is stable and it's totally fine, but the acrylamide monomer is pretty dangerous. What's interesting, and even though I know this, I still don't change my behavior with it. Acrylamide is oftentimes something that many of us might be ingesting ourselves without even realizing it. There's something called the Maillard reaction that happens in cooking that creates a browning effect on different meats when you cook them, but also for starchy dishes like potatoes, when cooked at really high temperature, like when you're frying them to make French fries, you'll get a reaction that produces acrylamide within French fries. And so if you are consuming French fries, you are consuming amounts of acrylamide, which is a neurotoxin and carcinogenic. So think about that next time you're having French fries. Though, again, I'm not practicing what I preach here. So I feel really bad if someone's eating French fries at the moment. So we discussed on the electrophoresis slide that when we run a protein sample across the gel, that's a native gel. What we can typically do is stain the native gel with a variety of different chemicals to try and identify, to answer specific questions. A really common one is called Kamasi blue. And it allows us, bless you, to, these different stains can let you look at total protein or phosphorylated protein typically. Kamasi blue enables you to look at total protein. So it's not specific to any proteins, it's just looking at, enabling you to look at bands that represent every single protein on your gel. And in the left lane here, so Cipro Ruby is another total protein stain. This is what the standard or lateral tells you. You know what the molecular weight is here for each of those bands. So typically what you'll is if you're looking for a specific protein at a specific molecular weight, and you're not interested in trying to use antibodies to detect that specific protein, but for example, if you have a bacteria colony that's expressing a certain protein and you just want to take what they're producing and see, like, should I see a thick band here designating that protein versus a normal bacteria that doesn't produce it, this is a helpful way to just quickly stain it and see on a native gel, are we getting our protein of interest? And again, this is looking at it basically at the by molecular weight for total protein. ProQ Diamond and PhosTag are two proprietary stains that look at phosphorylated protein. And so Kamasi blue, typically when you stain for it, it comes out blue, and it's actually something that comes from a chemical that comes from blue jeans. So before I move on, I want to take a step back and ask, I want to pause for a couple of questions here.
Could a gel ever be so like porous that like everything moves at the same speed?
That's a good question. So typically, when you're running a protein gel, you'll have there's a buffer that you'll have your samples in that will actually make it like visibly blue, and so you'll actually be able to see based on the voltage, like you're able to watch it run. And so like if you're just like running it through at a really high voltage, you might actually like mess up its ability to go through. So if the voltage is too low, like it's going to just like basically eke through and not make its way down. If you crank it up like 100, 120 volts, that's usually like standard. That'll enable you to like run it all the way down in about 30 to 45 minutes. And so the way these gels are made enable us to have separation based on, when it's SDSPAGE, separation based on size, where you'll have your really large molecular weight proteins towards the top and your smaller ones towards the bottom. Does that answer the question? If it didn't, you can say that, and that's okay.
No, I mean like hypothetically, could like
could they just like get stuck there?
no, like could it like because like it's separated off slack. Like could you make it such that like no matter the size, it just ran at the same speed? So that like all parameters.
Yeah, so hypothetically, yes. Like if you had a polyacrylamide network that was so porous that like everything just basically went through, that's hypothetically possible. But then I think also the stability of that gel would probably be pretty compromised and wouldn't work very well. But hypothetically, that's possible. Yes.
So you need both the native gel and the stain. What if you ran it without the stain? Wouldn't you be able to see the band?
Great question. You wouldn't see the bands like this.
Oh.
So there are other methods that you can do to look at and probe for proteins in your gel. If you're leaving your protein, this might sound weird, but if you're leaving the proteins in the native gel, typically what you can only do is some sort of stain. We're going to talk about a method next class where you'll transfer them to something else that allows you to use antibodies to probe for specific proteins.
But this is like a visual, you can see it in its own gel without further testing.
Exactly, yeah. All right. So I'm sure everyone is intimately familiar with this slide now, that every protein has an isoelectric point at which it reaches electrostatic equilibrium. What we can do is we can also separate proteins, as SDS-PAGE did it by size. We can also separate them exclusively by their charge. So while in class, we talked about this as a hypothetical situation where you have a tube of spectrum of pHs with your sample single protein here. There's a method that enables us that has a spectrum of pHs that, based on their isoelectric point, will be separated as a electric field is introduced across this spectrum of pHs, where they're separated based on their isoelectric point, based on their actual charge. And this method is typically called isoelectric focusing. And so it's a gel in which there's a gradient of pHs and there's an electric field applied across this gradient. It enables us to separate them based on their electric point. And typically what will happen, as we depicted previously with that previous slide in another class, as I just did now, the proteins will migrate through the gel until they reach a pH where they're uncharged. And so typically what will happen is the electric field will be at a lower voltage to enable them to slowly migrate through and find their isoelectric point, and at which time they're no longer going to be moving through at all. And the way what that typically looks like is, shoot, never mind. I thought I had it here. So there's a way to combine isoelectric focusing with SDS-PAGE, and we typically call that 2D electrophoresis. So it enables us to separate by both charge and by mass. What we'll first do is we'll do isoelectric focusing in a small cylindrical gel that separates them based on their charge. And you'll take that protein mixture that's separated by charge, and you'll actually take it and lay it on top of an acrylamide gel and do SDS-PAGE. So typically with SDS-PAGE, you have 10 wells and you'll have your pipette and you'll just pipette them in. Here what happens is the samples are separated out by charge already. You'll layer it on top and now just do regular SDS-PAGE to separate them by mass now. And the way what this will actually look like is this is a 2D gel, and we'll typically have a variety of proteins that are separated by molecular weight on the y-axis and their isoelectric point or their charge on the x-axis. And so one thing that's noteworthy is that there might be times where you see a blur. For example, protein 21, a lot of these are not all of them are clean circles or clean bands on this 2D gel. What ends up happening is, as we discussed in this class, proteins are altered all the time. They are glycosylated, they're phosphorylated, and so those modifications can change their charge or their size. We talked about in our ECM lecture how there are proteoglycans that have tons of sugar modifications, tons of GAGs on them that make them incredibly heavy. So the more glycosylation events that happen on a protein, the more sugars that are added to a protein increase its molecular weight. So you might see bands like, let me point it out over here. You might have bands like band 3 or band 4 that have a vertical tail on their on that protein, and that might indicate heavy glycosylation on that protein, or varying degrees of glycosylation on that protein. There's also horizontal smears on some of these proteins, which might indicate phosphorylation events or other kind of modifications that carry a charge with them. And so, again, as we described before, that the molecular weight for these proteins are typically reported in kilodaltons, and and daltons are typically grams per mole here. Before I step any further, are there any questions? Yes. You had it correct. Those are examples of why you might see smears. It's not exclusively glycosylation or phosphorylation. So like one possible explanation for like samples 3 and 4, why there appears to be a vertical smear, might be that that there's glycosylation events. It could be other things, because we don't know specifically what protein that is. So we don't know, typically in biology, certain proteins might have certain modifications. And so, we don't know what the modifications are on that protein, but one possible explanation could be like, those could be heavily glycosylated, or some of them might be. So like, the ones that are down on the bottom of that circle 3 or circle 4 might not be glycosylated, or sorry, the top. And then, forgive me, I'm twisted up today. The bottom might not be glycosylated. The heavier ones might be more glycosylated, or it could be some other modification that's changing their molecular weight just by a little bit. And over here, for like sample 21, is there another horizontal smear? 37 has a wide one. There might be modifications that are changing the charge of that protein. One possible modification is a phosphorylation event on that protein that'll change its charge. Does that make sense? Yeah, but again, those are not the exclusive ways by which that happens. Those are just possible explanations based on what we're seeing. Again, for a 2D gel, we don't know what this protein is yet. There's a way to try and figure that out. We'll talk about that next class. But for now, on this 2D gel, that's why they're labeled by number. We don't know what these proteins are yet. Yes?
So if isoelectrophoresis is going to like end every protein is reached like a neutral charge, but SDSPAGE requires multi-negative charge. How can you do one and then the other like in the same like gel?
It's a very good question. The best, so typically the SDS, okay, sorry. The SDS buffer that you use when running SDSPAGE in general is encasing the entire gel as well. And so when you separate based on charge, what's likely also going to happen is that there's also the buffer that it's in that'll also add a negative charge after they've already been separated. Typically, when we load these samples in an SDSPAGE, when you're just doing separating by mass, there's like other ways to try and further bombard them with a negative charge. But SDS is typically like this buffer, and like that you basically have your gel in this bucket that's like completely encased in SDS buffer. And so without having run a 2D gel myself, because I only ever do SDSPAGE, I suspect that what you're doing is after you lay it down and you have it on top of your page gel, you're basically enabling to bombard your samples with that negative charge because it's filled with that buffer. That's a really good question. I'm going to double check that. I'm pretty sure that's I'm pretty sure that's how you accomplish it. Yeah, that was a very good question. Yes.
So what about the dots that are like lighter at the top?
Are you talking about towards the bottom here,
So like, just,
or like over here where there's like dots that don't have a number on it?
just left.
Oh yeah, so that's likely a protein at low concentration or a low amount of protein there. This is just an example I pulled of a 2D gel. The fact if you see dots that don't have a number label on them, that's still a protein, but it's just an unlabeled protein. And again, we don't know the identities yet of those proteins when we run either a native gel with a Kamasi blue stain or a 2D gel like this. Yes.
Would it typically be tagged by computer, or would you like
Would it typically be what? Sorry.
tagged? Like, do we label the by hand or the computer?
So you would typically do it by hand if there's certain proteins at a certain molecular weight that you're interested in. So like, for example, like this, typically, I was answering a question earlier where when you're running your gel, you can see you have like there's a buffer that allows you to see your sample running down. But after it goes all the way through, your native gel, you don't see anything on it. It's a native gel looks blank. Once you apply some sort of stain like Kamasi blue, then you can see all of the proteins. So in this case, this 2D gel likely had either some sort of, not Kamasi blue here, but some sort of total protein stain that enables them to see everything, but yet not knowing what their identity is quite yet. And so it's tagged by hand after some sort of stain, particularly depending on where their molecular weight is, if you have something of interest. What likely happens here is, my guess what this person is doing is they've gone through and taken an image of their 2D gel and analyzed based on signal intensity to say anything that has a signal intensity above a certain threshold, call this a protein. Because, for example, as he was pointing out earlier, to the left, there's something next to the 14 here, but it's pretty, it's a little faint. That means it's a protein at a lower concentration and perhaps was below the threshold that they wanted to say, this is a protein to detect and identify. Does that answer your question?
Yes. So does it come to have artifacts? So maybe on proteins, or maybe like part of another group that just got left behind?
No, I wouldn't say, I wouldn't call them artifacts. I'd more likely call them proteins at low concentration that might look like an artifact, but are really just proteins at low concentration. Yeah.
Thanks.
Yeah. Okay. I'm going to say we're going to stop here and save the rest of this for next lecture to make sure that we have enough. So we'll go ahead and call it there.